All right, so get this. We're diving deep into mesenchymal stem cells, or MSCs as they're often called. They're like the body's little repair crew, able to morph into all sorts of cells, like bone, cartilage, you name it, even fat.
It's really quite remarkable.
But there's a twist. We're looking at how they function under a bit of stress, specifically low oxygen levels, which scientists call hypoxia.
Ah, yes, hypoxia.
You sent over some fascinating research on this. A 2015 article from Advanced Pharmaceutical Bulletin. It's called The Effect of Hypoxia on Mesenchymal Stem Cell Biology. So are you ready to explore how a little bit of stress might actually make these cells stronger?
Absolutely. I'm fascinated by this topic. What's particularly interesting is that hypoxia isn't always a bad thing. In fact, it's a natural part of their environment in the bone marrow.
That makes you wonder what we can learn by studying them in conditions that mimic their natural habitat.
Right?
What secrets might they reveal?
Exactly. And that's what makes this research so compelling. It's about understanding how these cells behave in the real world, so to speak,
Hmm.
and how we can potentially use that knowledge to develop more effective therapies.
Okay, so let's start with the basics. What are MSCs? Where do we find them? And what makes them so intriguing for researchers and medical professionals?
Well, MSCs are essentially a reserve of multipotent cells. They're found in various tissues, of course, bone marrow being a primary source, but also adipose tissue, umbilical cord, blood, even the placenta. And their claim to fame is their ability to differentiate. They're not limited to becoming just one type of cell.
So it's like having a cellular toolbox ready to repair and regenerate different tissues as needed.
Precisely.
That's pretty remarkable.
Yeah,
Now, hypoxia, most people associate low oxygen with something negative. But you're saying it's a natural state for MSCs in the bone marrow.
that's right. Think about it. Bone marrow is tucked away deep inside bones, not exactly a place teeming with oxygen. So for MSCs functioning under low oxygen is business as usual.
So exposing them to hypoxia in the lab is like giving them a taste of home, letting us see their true colors, so to speak.
Precisely. And that's where the research gets really intriguing. We see hypoxia influencing their behavior in various ways, sometimes even enhancing their abilities.
Hold on, enhancing. So a little stress is a good thing for these cells.
Yeah.
That goes against everything we think we know. What kind of effects are we talking about?
Well, let's take proliferation, for instance. Initially, hypoxia might slow down how quickly they multiply, but studies show that after an adjustment period, it can actually boost proliferation, leading to a greater number of cells.
So it's like they're acclimating to the challenge and then coming back stronger.
Exactly. And it doesn't stop there. Hypoxia also appears to influence what type of cell an MSC becomes their differentiation. Research suggests it might favor bone formation over fat.
Now that's fascinating. If we could manipulate oxygen levels to encourage bone formation, could that open the door to new treatments for bone fractures, osteoporosis, even things like bone loss in astronauts during space travel?
It's a very real possibility. And there's even evidence that hypoxia might protect MSCs from cell death. Both apoptosis, which is a programmed cell death, and necrosis, which is more of an uncontrolled cell death due to injury. It seems a little stress can make them tougher.
Wow. So not only are they multiplying more and potentially becoming bone cells, but they're also better at surviving. It sounds like they're truly thriving under these conditions. What about their metabolism? How do they fuel themselves in this low oxygen environment?
That's another remarkable adaptation. Hypoxia forces them to become more energy efficient, shifting their metabolism away from processes that rely heavily on oxygen.
Okay, so they're adapting their energy production, they're surviving better, they're potentially differentiating into bone cells more effectively. What's the driving force behind all these changes? Is there some kind of control center orchestrating this response to hypoxia?
There is. It's a master regulator called HIF, or hypoxia-inducible factor.
Mhm.
Think of it as a switch that gets flipped when oxygen levels drop. This triggers a cascade of changes at the genetic level, leading to all the adaptations we're seeing.
So HIF is basically sensing the low oxygen and telling the cell, all right, time to activate plan B, let's adapt and thrive.
Precisely. And what's incredible is that HIF regulates over 1,000 genes, fine-tuning the cell's response to hypoxia. It's a complex and powerful system.
This is all so mind-blowing. We've got these remarkable cells, the MSCs, responding to low oxygen levels in ways that could revolutionize medicine. We've barely scratched the surface and I'm already hooked.
And there's so much more to uncover. We haven't even touched on how hypoxia affects their ability to migrate,
Right.
which is crucial for them to reach areas in need of repair.
Well, I don't know about you, but I am ready to dive deeper into this. Let's take a closer look at some of the specific research findings and what they might mean for potential therapies.
Welcome back. Let's delve into some specifics from the research on how hypoxia really impacts MSCs.
All right, so you talked about hypoxia potentially boosting how fast MSCs multiply. What did the researchers actually observe in the lab?
Well, they took umbilical cord-derived MSCs and cultured them under different oxygen levels. They found that at 2.5% and 5% oxygen, significantly lower than normal conditions, the cells actually proliferated more than those in a typical oxygen-rich environment.
Wow, so less oxygen actually led to more cells. That's pretty counterintuitive. Did the researchers notice any changes in the cell's appearance or characteristics? What scientists call phenotype under these lower oxygen levels?
Yes, they did. In one study, they compared MSCs grown under 5% oxygen, which is similar to their natural bone marrow environment, to those grown under normal oxygen conditions. And interestingly, the MSCs in hypoxia initially grew slower. It was like they were taking their time to adjust to the environment.
Like they were getting acclimated to their new digs.
Exactly. But then, get this, in subsequent passages, they actually developed a growth advantage. It seems like that initial adjustment period made them more robust in the long run.
So they were playing the long game, adapting to become stronger. Fascinating. What about those unique surface markers that identify MSCs? Did those change under hypoxia?
That's a great question. And thankfully, the answer is no. MSCs, regardless of whether they were cultured under normal or hypoxic conditions, consistently expressed those key surface markers. Things like CD73, CD54, CD44, and CD90, all present and accounted for.
So the identity of these cells as determined by those markers remains stable even in low oxygen. That's good news for researchers and clinicians who need to be able to reliably identify these cells for therapeutic use. Speaking of therapies, you mentioned that hypoxia might favor bone formation. What kind of evidence did they find to support that?
Well, they looked at specific markers that indicate bone formation, one of which is called ALPL. They found that ALPL expression was significantly higher in MSCs cultured under severe hypoxia compared to normal oxygen conditions.
Okay, so that's a pretty clear sign that those cells were leaning towards becoming bone cells. What about markers for fat formation? Did those show a decrease under hypoxia?
Yes, they did. They looked at a marker called PPRG, which is a key regulator for fat formation. Interestingly, PPRG expression was much higher under normal oxygen conditions compared to hypoxia.
So hypoxia seems to be tipping the scales, encouraging bone formation and suppressing fat formation. What are the implications of this finding for potential therapies?
This could be a major breakthrough for bone regeneration therapies. If we can manipulate oxygen levels to push MSCs towards a bone-building pathway, it could lead to new treatments for a whole range of conditions, from fractures and osteoporosis to even bone loss in astronauts during space travel. Imagine a simple tweak of oxygen levels could help heal bones faster and more effectively.
That's incredible. We're essentially learning how to nudge these cells toward a specific fate by tweaking their environment. You also mentioned that hypoxia might protect MSCs from cell death, which sounds counterintuitive. What did the researchers discover about that?
Researchers found that when they cultured those umbilical cord-derived human MSCs under very low oxygen, just 1.5%, there was a slight increase in apoptosis, that programmed cell death we talked about earlier.
Wait, I'm confused. I thought you said hypoxia was protective.
It is in a way. While apoptosis increased slightly, the important finding is that the overall levels of cell injury and necrosis were significantly lower in those hypoxic conditions compared to the control group with normal oxygen levels.
So it's not that hypoxia prevents all cell death, but it seems to shift the balance towards a more controlled form of cell death. Is that because of how they adjust their energy use under hypoxia?
You got it. Under those extremely low oxygen levels, the MSCs consumed more glucose and produced more lactate compared to normal oxygen conditions. This suggests they shifted to a more anaerobic or oxygen-independent way of generating energy.
So they were basically adapting to the lack of oxygen by switching to a different energy source? That's remarkable. And does this metabolic shift play a role in their survival under those challenging conditions?
Absolutely. This metabolic adaptation likely contributes to their resilience, making them more efficient at using available resources.
It's incredible how these tiny cells can reprogram themselves to survive under stress. And remember, HIF, our master regulator, is it involved in orchestrating this metabolic shift as well?
It is. Studies show that hypoxia leads to an increase in the expression of genes involved in energy metabolism, such as GLUT1, LDHA, and PGK1. And guess what? HIF regulates all of these genes. It's like a conductor leading an orchestra, making sure everything works in harmony to keep the cells alive and functioning.
It's amazing how one factor can control so many different processes within the cell. Now, we haven't talked about migration yet, the cell's ability to move around the body. This is crucial for MSCs to reach areas in need of repair, right? What did the research reveal about hypoxia's impact on their mobility?
One fascinating study showed that hypoxia actually triggered MSCs to move from the bone marrow into the bloodstream. They essentially mobilized into circulation. ready to be dispatched to sites of injury or damage.
So hypoxia acts like a call to action, mobilizing these repair crews to address issues throughout the body.
That's one way to think about it. And intriguingly, this mobilization didn't seem to deplete the bone marrow's supply of MSCs. This suggests that there might be other sources of these cells that contribute to the circulating pool, which it could be very good news for developing cell-based therapies.
So we may have a larger reserve of these cellular repair units than we previously thought. That's exciting. But this migration can't be random, right? How do these cells know where to go?
You're right, it's not random. They follow specific signals like what's called the SDF1-CXCR4 pathway, which plays a critical role in stem cell recruitment. It's like they're following a homing beacon to their target destination.
So they're not just wandering aimlessly, they're being guided to where they're needed most. What other factors influence their ability to navigate through the body?
Metalloproteinases are another key player. These enzymes help them break down barriers in the extracellular matrix, essentially allowing them to move through tissues more easily.
It's like they have their own little toolkit for navigating the complex terrain of the body. Does hypoxia affect their expression of these metalloproteinases?
Yes, it does. Hypoxia can actually influence the expression of these factors, fine-tuning their migration abilities and ensuring they can reach their destination.
This is all so intricately connected. It's really remarkable how these cells respond to challenges and adapt to their environment. I'm eager to learn more about how this new understanding of hypoxia and MSCs can be translated into actual therapies.
Let's explore those exciting possibilities in our final segment.
Welcome back to our deep dive into hypoxia and mesenchymal stem cells. You know, I'm already amazed by how much we've learned about these adaptable little cells.
Yeah, it really highlights the power of basic research. Discovering these intricate mechanisms at the cellular level could lead to some incredible breakthroughs in medicine.
Absolutely. And that's what I'm most excited about, the potential to translate all this knowledge into therapies that can actually help people. So where do we go from here? How could understanding hypoxia's effects on MSCs lead to better treatments?
Well, we know hypoxia can boost bone formation and enhance MSC survival. What if we could leverage that knowledge? Imagine preconditioning these cells with low oxygen levels before transplanting them into a patient.
So it's like giving them a boot camp in the lab, prepping them to be stronger and more effective once they're introduced into the body. I love that idea.
Exactly. This preconditioning could dramatically improve the success rate of stem cell therapies for conditions like bone fractures, osteoporosis, even cartilage repair.
Instead of just hoping for the best, we're giving these cells an advantage right from the start. What other therapeutic avenues are researchers exploring with this new understanding of hypoxia and MSCs?
One area that's particularly intriguing, though it might seem counterintuitive at first, is cancer treatment.
Wait, cancer? But don't tumors thrive in low oxygen environments? How could hypoxia help us fight cancer?
That's a great question. You're right, tumors often develop in hypoxic conditions, but researchers are exploring ways to manipulate that hypoxic environment to make tumors more vulnerable to therapies like chemotherapy or radiation.
So we're not just using hypoxia to enhance the MSCs themselves, but also to change the playing field within the tumor itself, making traditional treatments more effective.
Precisely. It's a two-pronged approach. And MSCs could even play a role in delivering those therapies directly to the tumor site, acting as a sort of guided missile.
That's remarkable. We're essentially using the tumor's own environment against it. What other diseases could potentially be targeted with MSC-based therapies enhanced by our understanding of hypoxia?
The possibilities are vast. We're talking about heart disease, stroke, spinal cord injuries, autoimmune disorders, the list goes on. Imagine using MSCs to repair damaged heart tissue after a heart attack, or even to help paralyzed patients regain movement after a spinal cord injury.
It's mind-boggling to think that these tiny cells, once a scientific curiosity, could hold the key to treating so many debilitating conditions. This research is giving us hope for treatments we could only dream of before.
Absolutely. And it all stems from understanding how these cells function in their natural environment, responding to challenges like hypoxia in ways that we're only beginning to appreciate.
So, as we wrap up our deep dive, what's the key takeaway you want our listeners to walk away with? What should they be pondering after learning about hypoxia and mesenchymal stem cells?
I think the biggest takeaway is that stress in the right context can be a powerful driver of adaptation and even enhancement. We've seen how hypoxia, a seemingly negative condition, can actually make MSCs more resilient, mobile, and effective. It's a reminder that sometimes a little bit of adversity can make us stronger, even at the cellular level.
It's like that saying, what doesn't kill you makes you stronger. And in this case, it might just lead to groundbreaking new therapies.
Exactly. This research also underscores the importance of understanding the natural environment of cells.
Mm.
We can't truly unlock their potential unless we study them in conditions that mimic their natural habitat.
It's like trying to study a deep-sea creature in a shallow tide pool. You won't see its true capabilities unless you observe it in its natural environment.
That's a great analogy. And that's precisely what researchers are doing with MSCs and hypoxia, observing them in their element to unlock their therapeutic power.
Well, this has been an incredible journey into the microscopic world of cells under stress. I feel like I've gained a whole new appreciation for the resilience and adaptability of life itself.
Me too. It's exciting to think about what the future holds as we continue to explore this fascinating interplay between hypoxia and mesenchymal stem cells.
To our listener, we encourage you to delve deeper into this incredible field. The Advanced Pharmaceutical Bulletin article is just a starting point. There's a whole world of knowledge waiting to be discovered, and who knows, maybe you'll be the one to make the next big breakthrough. Thanks for joining us on this deep dive, and until next time, keep exploring.